Yes, yes.
Yeah.
Okay. So it's a pleasure to welcome here Mr. Bobby Ranjan. It's Mr. for only 6 months, so then you will discuss your PhD. So, Bobby Ranjan has studied in Singapore together with Chaitanya Joshi, and you have got your bachelor at Nanyang University in Singapore, and then you have worked as a bioinformatics specialist at the Singapore Genome Center, which is a very is a fantastic institution because it has made a lot of achievement in genome science. And then you started the PhD in Jamie Hackett group in MBL Rome, and it is really something very, very cool, because it's about epigenetics, and epigenetics is about everything in our life, how we age and so on, and also about microbiome, and microbiome is in some sense the other ego of us, the other self, and everything that is we try to forget, but there is. And this kind of, we will talk today about this kind of link between epigenetic and microbiome, particularly with respect to the disease risk. Yeah. Thank you very much.
Thank you very much for the introduction. As you heard, I'm Bobby, and I'm getting towards the end of my PhD at EMBL Rome. And today I'm going to talk about microbiome-mediated inheritance, primarily epigenetic inheritance of disease risk. But I want to start my introduction with a concept that's familiar to most, starting with Mendelian inheritance of what we've looked at in biological textbooks, or even nowadays in statistical textbooks, of inheritance of traits where you have two heterozygous parents and a gene with two alleles where the dominant allele shows complete dominance over the recessive allele. And what you can see is there's three possible outcomes with different probabilities, the heterozygote having a 50% probability and the recessive and dominant having 25% probability each. And I want to use this as the basis for a thought experiment to ask the question, what if we have a trait where along the x-axis you go from a high value on the right to a low value on the left. And the idea is if we have this trait determined by a single gene with two alleles, assuming complete dominance, there's three possible outcomes, as I showed before, with different population frequencies. And you have your genotypes on the x-axis going across different values of the trait. If I say this trait is now determined by two genes, you now have 5 possible outcomes determined by 9 possible genotypes. If it's 3 genes, you now have 7 possible outcomes. 4 genes, you have 9 possible outcomes, and so on. You can go up to 10 genes, you can keep going further and further. What's interesting to show is that this distribution can be modeled using binomial coefficients. So essentially what you're doing is you're choosing out of 2n possible alleles, different numbers of dominant alleles across this distribution. So you can choose on the extreme left, no allele is dominant, then 1 allele is dominant, 2 alleles are dominant, and so on. You can spread this distribution all the way to all alleles are dominant. So 2n choose 2n, which is 1. You can compute the mean and the standard deviation of this distribution, and you can also show that this distribution tends to approach normality as you increase the number of genes that determine this trait. What this means is that given your genotype and given your genotype of your partner, I can predict with reasonable accuracy the probability of inheritance of this trait and the position of the person, of each individual person along this distribution. However, multifactorial disease risk is not inherited in this manner. And in the 1960s, a concept of liability was proposed, wherein different people, different groups of people, different groups of animals were positioned along different positions of this distribution. And essentially, the incidence of disease was modeled as a threshold beyond which the disease was onset. So what happens is in this distribution, you have a fixed position for when the disease happens, called the beyond which you measure incidence, and different groups of people sit on different distributions. And what you can see is the distribution below, the incidence is about 20% of this particular disease, whereas in the distribution above, the incidence is about 5%. This example here is between male and females, but this could be across different ages, this could be across different countries of origin, and so on. And this predisposition, this liability, is determined clearly not exclusively by genetic traits, but also includes non-genetic factors that contribute to this liability of multifactorial disease. Non-genetic factors is a very broad term and includes a whole host of possible exposures, going from epigenetic information in the gametes to in utero exposures to possible postnatal environments. And in our lab, we focus on epigenetic information in the gametes. And this specifically means looking at non-coding RNAs, looking at methylation of the DNA, and finally looking at modifications and possibly histones. And there are different studies across many species that have shown this. A couple of examples of this are C. elegans, where there's an interesting study on the inheritance of attractiveness in worms and how, based on the small RNA composition, different worms are more likely or more attractive to their possible mates. In mice, on the other hand, behavior and response to stimuli has been shown to be inherited from parents to offspring. And very famously, the coat color has been shown to be inherited as an epigenetic mechanism. Even in humans, sperm has been shown to carry epigenetic signatures of environmental exposure, like cigarette smoking, for instance, which has been shown to modify DNA methylation signatures, which then has affected the offspring DNA methylation. So in general, this sets the precedent that the paternal environmental exposure can impact the offspring, potentially through molecular information in the sperm. Given this, we wanted to look at a system in mammalians, in mammals, that could model paternal environmental exposures and propagate that to the next generation. We know from decades of research that the gut microbiome is sensitive to environmental exposures, to drugs, to the temperature outside, to the diet we consume, and so on. And we also know, more recently, that the gut microbiome propagates this information to various somatic tissues in the body, starting from the brain to the liver, lungs, and so on. So very simply, we asked, can this influence extend to the germline? Can we measure information that is propagated from the gut microbiome to the germline? This communication we call the gut-germline axis. To actually go out and discover the gut-germline axis, this project was started by a former postdoc in our lab, Ayele, who later on has gone to start his own lab in Freiburg. And he started with a simple experiment of giving non-absorbable antibiotics to male mice. So these antibiotics are ingested, they enter the body, they clean out the microbiome and are excreted. They don't enter the bloodstream, which is important because there is no direct effect of the antibiotics on the somatic tissue. The effect of the antibiotics is through the microbiome. And what we saw when we mated these antibiotics-treated males with conventional females is that there is a drop in the survival of offspring over about 2 months of post-birth. Ayele also modeled this using alternative ways of perturbing the gut microbiome, using an alternative set of antibiotics, and using a laxative that does not necessarily mimic the antibiotic properties, but still cleans out the gut. I want to introduce a concept before we go ahead of birth weight being a predictor of disease risk. And as you can see from the pixels on this figure, this is an ancient concept that we know for a while now that birth weight is very predictive of especially onset of metabolic disease, type 2 diabetes, and so on, and heart disease. And also there is a risk of infant mortality if the birth weight is extremely low. What we found from the antibiotics treatment model is that there was a significant drop in the birth weight of offspring coming from antibiotics-treated fathers. The effect was propagated throughout the early days of development until weaning, at least. And at every time point, we see a significant drop in offspring birth weight, both in the males and the female offspring coming from antibiotics-treated fathers. Combining these two, combining the male and female offspring, we see that at, let's say, pre-weaning at P15, we see a significant drop in the offspring body weight, which can be modeled also as a distribution by binning, standardizing, and z-scoring these weights. And just to give you a visual image of what this looks like, the conventionals or the normal offspring are on top. The offspring of antibiotics-treated fathers are at the bottom, and you can see there's a clear difference in the size and even the amount of hair on the bodies of these offspring. And this is exclusively from giving the fathers antibiotics. In addition, we see that there is an increased likelihood of severe growth restriction if we bin these birth weights by standard deviation. And not only is this upon antibiotics treatment, but also we see this when you give alternative antibiotics and the laxatives, that we mimic a severe growth restriction phenotype in the offspring. And the rescue of the microbiome, very interestingly, rescues the offspring phenotype. So if we wait for about 4 weeks, we still see the phenotype of offspring mortality. But if we wait up to 8 weeks where we see the microbiome has fully recovered, we can see that the offspring phenotype is also rescued. And this is not just upon survival, but also upon in the growth restriction and birth weight phenotype. So all of this put together indicates that giving the father antibiotics, essentially paternal gut dysbiosis, is essentially lacking in some information that's being passed on to the offspring, which is resulting in an increased mortality and birth weight associated phenotype. But we're not exactly sure what the information is that's being passed. And information transfer from the male to the female can occur through multiple different mediums. Now, the assumption is that this information goes through the sperm and seminal fluid, but they could also have transfer of saliva, fecal matter, because mice are coprophagic, and social and behavioral cues. And to disentangle the germline-specific information, we turn to in vitro fertilization, which very simply involves stimulating and taking out the eggs and the sperm and performing an in vitro fertilization assay, culturing the embryos in vitro, and implanting the embryos back into the mother. What Ayala found when he did IVF is he found a similar F1 birth weight discordance, and this is the schematic of the experiment he did with essentially oocytes coming from equal pools, let's say, going to antibiotics-treated sperm from antibiotics-treated males and conventional males. And using 2 different surrogates, he found that he could reproduce the birth weight phenotype in terms of the birth weight of the offspring coming from the antibiotics-treated fathers were lower than the conventionals. So I'll take a bit of a break here on this story, but there's a lot more in this paper that came out about a year ago that reveals that there is an axis of communication between the gut microbiome and the germline. Perturbing this axis through multiple approaches impacts offspring survival, birth weight, and body weight phenotypes. And this intergenerational phenotype through IVF indicates that there is a germline or germline-associated information transfer that is communicating paternal gut dysbiosis to the next generation. Finally, the restoration of this paternal gut microbiota rescues the offspring's phenotype. This attributes a causal role to the gut microbiome in affecting the offspring development. Now, I want to stay on the track of IVF and mention that this also allows us to culture embryos in vitro and study the information in early embryonic development that might be passed from the father to the offspring. Because we have these embryos in culture in the lab, and essentially what we can do is from the zygote, culture these embryos all the way to the last stage of pre-implantation development, which is the stage before which we implant the embryos into the mothers. We wanted to see if we could capture information of paternal environment even before implantation and thus potentially predict the offspring phenotype. For this, together with Mathilde, former postdoc in the lab, I led the story, co-led the story on understanding essentially multi-paradigm paternal effects on the next generation. We have two, let's say, perturbation models and a control, wherein one of them is the aforementioned antibiotics model, and another model which is studied by our collaborator Anna Boskovich's lab, which is a low-protein, high-sugar diet, or a diet-specific perturbation. Using the IVF model I just described, Mathilde collected sperm from all of these males across different environmental conditions, performed IVF and cultured the embryos until blastocysts, and performed single embryo transcriptomics to essentially sequence the RNA from each individual embryo and give us a distribution of states, of embryonic states coming from the different fathers. This allows us to capture essentially information at the earliest possible stage of whether, let's say, the offspring is affected by the paternal treatment, and to understand also how it is affected. Using very simple linear principal component analysis, we already find hints of a global separation. If you look at the histogram on the top of the PCA, you can see that a lot of the controls tend to cluster around the center of the distribution, and the two conditions tend to split on either side of the controls. Based on differential expression, we can capture some signatures. Very broadly, I'll go over the fact that on the diet perturbation, we see a more metabolic or metabolism-associated phenotype. And on the antibiotic-treated model, we see a more developmental and differentiation-associated phenotype. And I want to use the study to talk about something very relevant, which is sources of variation in this epigenetic inheritance paradigm. What we observed is that different batches of different antibiotics, treatments, or different diets, and so on, can have slightly different responses at the embryonic level. And so what Mathilde did was to perform same-day IVFs across the three conditions so that we could have comparative analyses, minimizing as much as possible technical effects. And from the PCA before, what you can see is there is batch-specific separation. So we do need to analyze individual batches one by one, or correct for this batch effect to somehow get to a consensus understanding of what's happening in the embryo. And we did the first approach, which is analyze each batch individually. And on the left, you see the results of the antibiotics treatment model, on the right, the low protein model. And you can see that different batches show different extents of separation between the control and the treated embryos, let's say. And essentially, despite this batch-specific variation, we were able to capture some consistent gene expression changes. Specifically interesting is GATA4, which is an early embryo marker of lineage specification. I wanted to also point out that we tried to look at this data in a slightly different manner by quantifying variation in transcriptomic in blastocyst development, looking at the coefficient of variation. So the idea here was to essentially fit a generative additive model to our data and to the distribution of the coefficient of variation and mean to look at what is the relative variation of a gene's expression in a particular data set relative to what we would expect from a fit. And we tried this to compare two different genetic backgrounds, and we found that actually embryonic gene expression is more variable in the Black 6 background than in the FAB. These are two different genetic backgrounds of mice. And the idea was to understand, can we use this as a model for studying embryonic development? If we compare the variability of genes in the different genetic backgrounds to look for treatment-specific signatures, we do find genes that are more variable than expected in both genetic backgrounds that is common to the paternal treatment. And of particular interest on the low protein side was the Mervel-associated Mervel2A element, which has been previously implicated in low protein diet-associated phenotypes. So from this, essentially, I wanted to say that there are consistent reproducible gene expression changes that we can capture across batches in early embryos and that are specific to paternal exposure. We do need to be careful about batch-specific variation, and especially when we use linear approaches like principal component analysis to summarize our data. And we tried a different approach with the variability-based analysis that provides an orthogonal method for studying gene expression changes in such data sets. And we think we can use this to tease out some effects of embryonic priming that might implicate postnatal effects. We have summarized all of this work and more in a recent preprint that came out a few months ago, and so feel free to go through that. And I want to close this part of the talk and acknowledge many people, including Jamie, Ayala, and Mathil, primarily who've helped me shape my way of scientific thinking over the last 4 years. The Mouse House, without whom any of this would be possible, specifically Danilo and Violetta, and Anna's lab for collaboration, Yanning for behavioral stuff, which I haven't presented here. And before I go, I want to give you a quick overview of my background and future plans. And as Pietro introduced, I did do my bachelor's in computer engineering at Nanyang Technological University, from where I moved to the Genome Institute under Shyam Prabhakar as a bioinformatics specialist. What I was interested in there was developing methods for single-cell transcriptomics analysis, and we came up with a few different methods for clustering and feature selection in single-cell transcriptomics. I then moved to Rome, to Monterotondo, more specifically with Jamie Hackett, where we studied the intergenerational effects of gut germline communication, primarily the microbiome-related perturbations I showed you, and the multi-paradigm study on the protein diet and the antibiotics treatment. What I also have done along this is teaching mathematics to our bioinformatics community, including a few lectures on eigenvalue decomposition and ending with linear algebra, concepts of linear algebra. And this year I want to go into more nonlinear concepts. And so I wanted to tease a little bit, give you a little bit of a teaser on what's next and what I would like to do going forward, primarily on understanding epigenetic inheritance, but the challenges from what I've shown you so far, especially the linear methods like PCA, struggle to handle the complexity and the variation in data sets, especially of epigenetic inheritance. And I think, in addition, what we need is integration of different environmental perturbations and potentially different species to understand better the landscape of epigenetic inheritance. More recently, some deep learning approaches have been successfully applied to gene expression analysis, primarily focused on variational inference, where they've looked at essentially decoding and encoding single-cell data sets into a latent space. What is, unfortunately, a lot of these models are developed for a general purpose, unperturbed wild-type single-cell data set, and they're primarily focused on, let's say, immune cells or easy to access tissues. The models that are for mouse and human embryos, for instance, the one in the middle here more recently, has taken the variational inference approach and added a deep explainer to it, so you can understand contributions of different features to predictions of cell types and cell states or manifold alignment. The challenges and what I would like to do going forward is to develop such bespoke deep learning approaches to study epigenetic inheritance across species, because we can clearly see there is successful application of deep learning methods in this domain, but the perturbations and let's say the spread of different epigenetic paradigms is difficult to access. With this, I want to close, but I'm happy to chat about these results and my future plans over any form of food or beverage. And I want to thank you for your attention, and happy to take questions.
Yeah, I mean, if anyone has questions, they should be free to ask. Also, so for people online, like you can mention, and they may not hear me, but they
Uh-huh.
can mention.
Yeah. So people online, feel free to ask questions. I think you can drop a comment in the comment box, or you can also, I guess they can unmute themselves as well. So.
Yeah. And I had a question.
Yeah.
So towards the end, you talked about existing work on deep learning and related to doing things with gene expression. Why do you think very little work has been done on epigenetics here? Like, is it a data issue? You know, like what do you think is more challenging? Is it like the data sets and that kind of work, or the modeling work?
I think it's a combination. I would say experimental design, more specifically, which is sort of a combination of both of these concepts, because the question of studying epigenetic inheritance at this resolution involves designing the experiment a priori with the objective of generating these large data sets with a lot of both throughput in terms of cells, but also throughput in terms of modalities and resolution of your data set. And then being able to take that and learn different nonlinear manifolds of how this data, let's say, what is the shape of this data set. I think the combination of that and the design of the experiment in such a situation has been challenging. And I think this is a good time to jump into this question. Yeah.
So I'm aware of some optimal transport methods,
Yep.
which kind of map from between two different manifolds.
Yep.
I'm currently looking at a data set which will do that.
Mm-hmm.
And I was wondering if there is any work that takes the ataxic data,
Mm-hmm.
which I think is chromatin data,
Yep.
maps it on how it changes the gene expression of cells.
Yep.
And one of the things that I'm looking for is a time varying version.
Mm-hmm.
And it's just very difficult.
I agree, 100%. Mapping ataxic, especially single cell ataxic, onto gene expression is kind of an open question at this point. The initial, let's say, approaches with, for instance, the CRAT packages was to use a canonical correlation analysis, which is still assuming linearity between two spaces and sort of projecting the ataxic space onto the RNA-seq space, and then correlating changes in chromatin accessibility with changes in gene expression. I still think we need different ways of thinking. Optimal transport is probably one way to do that. But you have to, for instance, to validate whether such an approach works, I think it is important to have, let's say, ground truth data sets. I don't think we have that yet. The single cell multi-ohm assay where you can get ATAC and RNA from the same cell is a step towards that. But I still think a lot more, let's say, targeted molecular biology needs to be done to actually validate that changes in accessibility in certain chromatin states actually affect gene expression.
What do you think of the amount of noise in the data? Because I was working with something which had both chromatin data and gene expression data.
Mm-hmm.
It was just extremely noisy. And methods that did work on similar data sets don't necessarily the deep learning methods don't consider.
I agree.
Noisy ones.
Yep.
So we then tried a bunch of things, smoothing, or we tried different sampling techniques, but it just doesn't work because of the noise.
Yep.
So do you have any insights on that?
Yeah, definitely. I think a lot of the noise we see in epigenetic inheritance especially, some of it, I think, is a lack of analytical tools rather than actual experimental technical noise. For instance, even modeling partial penetrance of a phenotype, we don't really have ways to do this at a transcriptomic or a genomic level. We don't really have probabilistic modeling in a nonlinear fashion. And the methods that do, for instance, there are some variational inference methods, which is, I think, SCVI, which came out a few years ago, and now it's been adapted for cell type annotation. It does work with that concept of probabilistic modeling of cell states, but of course, it remains to be seen if that concept will apply to different epigenetic inheritance paradigms. Yeah.
does it relate human beings, like in the sense of translation of the data?
Yeah, that is something I am interested in my, as kind of I was planning to do at the end of my PhD and might do going forward, which is studying the human microbiome and trying to predict offspring phenotypes from human data. And actually, part of the reason I'm here in Cambridge is there is a course at EBI that is specifically focused on studying human cohort data. And I am interested in accessing data from multi-generational cohorts, getting microbiome data from the parents and seeing if certain bacterial strains or even combination of strains are predictive of offspring outcome. So it remains to be seen how well we can model that data, firstly, and secondly, how predictive it is of offspring outcome. But the signs are good, at least from mouse work, and I'm curious to see how well it translates. Yeah.
Question is, since you are modeling based on bad behaviors and like in terms of antibiotics that the mice has taken, is there something that you guys tested conversely opposite? Like let's say if they have good behavior or like good dietary, let's say high protein, high fiber, you know, like those good habits, so-called like the offspring will be like way better.
Yep, I am just writing up that paper. So in a study which I haven't presented here, we take germ-free mice. So these mice start off in isolation with an absence of in completely sterile environments. And then we put back different strains of bacteria, including a positive control from a normal mouse. And the idea is to see what is the theoretical limit of microbiome perturbation. On a positive end, can we put back the microbiome and predict and essentially rescue the germ-free mouse and make it look like a conventional mouse? So this is, I'm actually just writing up this paper. So maybe next time I can show you all of it.
There's a question online which I will read out.
Sure, thanks.
So Ranjan asks, are there any specific types of microbiomes or bacteria in the gut that are known to have substantial influence on offspring? So I'm wondering if there's a reliable method to profile microbiome composition and whether this could bring insight.
To take your first question, no, we don't know if specific strains are more or less predictive of offspring outcome, but that is part of the story I am working on and trying to understand just the dynamics, what are the principles of communication between the gut microbiome and the germline. And it is a big question, so I don't think I will answer this question, but I can try to guess if specific bacteria are more or less relevant for offspring outcome. For the second question, we're quite good with measuring, let's say, the quantification and both of abundance and composition of the microbiome, both in humans and mice and so on. The challenge is, of course, understanding whether they're individually functionally relevant in terms of the bacterial strain itself, whether the genes in the bacteria that are communicating with one another are modifying their function going downstream, or if, for instance, their presence just in terms of abundance affects the somatic and maybe the germline going forward, and maybe they're not actually functionally doing anything, but their presence modifies the host cells around them and allows for germline communication. That question is still open, and so hopefully in a few years we have some insights into that.
I had another question. So we're at the CS department, and a lot of R people, deep learning people have been really excited about AI-driven virtual cells. Generally, like modeling, let's say, like cell perturbations with like foundation models.
Yeah.
I'd love to like hear your thoughts about it as someone who's worked with experiments, who's worked with gene expression data, population as well.
Yeah. I think we're still not there yet with the modeling of the virtual cell. I think there is still a lot more experimental work to be done to understand, firstly, what are the components involved in the epigenetic landscape of a cell, and then to take that into inheritance to the next generation. I still think from a foundational model perspective, we can learn a lot from first principles from biophysics, for instance, the way proteins bind to DNA and just physical forces. There is a lot we can learn about epigenetic gene regulation. But I think in terms of really understanding an entire cellular context from first principles, I don't think we are there yet. Yeah.
I just want to understand from an experimental point of view, how much time and effort does it take to produce, let's say, any kind of data?
Yeah.
Like,
It is.
let's say I want a dynamic data. I want to understand how the chromatin peaks are affecting the gene expression with, like, let's say, 12 days or something.
Okay.
How much time and effort will it actually take?
It can be months of work. So just in terms of the organization, because firstly, for instance, your question would be context specific. If you're asking whether chromatin peaks affect or how chromatin peaks affect gene expression, the question is in what context? Is it in embryonic development? Is it in germ cell differentiation? Is it in, I don't know, liver cells, for instance? Because they all have different epigenetic concepts. Is it in cancer, where it is epigenetics is very interesting. So once you fix your context, you then want to find a model, whether in vitro, in vivo, or so on. And if you then decide to go with, let's say, an in vivo model like mice, you then want to get the mice to an age or a model that is potentially genetically modified or exposed to an environment that induces changes in the chromatin accessibility that you're interested in, and then propagates that, and then allows you to profile gene expression there. What's very challenging with mouse work is being specific with the perturbation, because, for instance, you want all of the systems in many ways are linked to one another. So if you perturb something, it's unlikely that you specifically perturb one system. So you're going to have feedback loops and effects across the system, primarily immune, usually with toxicity-based effects. And that can communicate to the brain, that can communicate to the digestive system, and so on. And suddenly then you have nutritional issues, you have differences in hormonal profiles, and then that comes back to the organ you're interested in. So I would say that it is a very complex question. And it's more than actually the time in the lab to actually do the experiment. What the biggest challenge with answering a question like that is, can you narrow down your context enough that you're still able to answer the question in a feasible manner that takes a finite amount of time? Because these questions can blow up into massive projects here.
So I can ask that question.
Sure.
So based on your experiments, it's like what time frame did you actually process, like removing like the previous dietary or microbiome that the mouse has to actually produce this genotype that you would ideally want, and like the sampling size of the mouse, like let's say ages this and that, and to actually what you ideally kind of want within the lab space.
So what we, let's say the important time points we selected were basically the duration of the antibiotics treatment, which was about 6 weeks, because one cycle of spermatogenesis in mice takes about 5 weeks. So the idea was we want to treat the mice for longer than one cycle, so it's very likely that the sperm we collect for IVF, or even the sperm that goes into natural mating to produce the offspring, are sperm that are at some stage affected by the antibiotics treatment. And and it's unlikely we get sperm that are completely unaffected by the treatment. So that was an important time point. We started the treatment at about 5 weeks of age, which is post-weaning, and at a time point where we can say the mouse are sexually mature, more or less, and then continued for 6 weeks. So we stopped the treatment at 11 weeks. So those were the decisions we made in terms of time points of the antibiotics treatment, and we did a similar thing for the diet as well. Great.
Now, lot of people said, nice presentation.
Thank you.
And yeah, thank you for a great presentation. We can chat afterwards. Thanks everyone online as well. And we're going to end the meeting now. Okay. Just going to make sure that the recording is saved.